Hi, my name is Alani Martinez. I just graduated from Kova High School and I am an incoming freshman at U of A. This summer I spent studying under Dr. Jonathan Sanchez at the Mycona lab, and our research focused on Alzheimer's. The lab's main question was, how can we inhibit necroptosis? So the reason our lab wants to answer this main question is trying to prevent necroptosis from happening and forcing the cell to either go into apoptosis or cell survival. So our research and experiments that our lab conducted were trying to find a possible compound to prevent two proteins from binding together and leading to a stop of the necroptosis pathway. The work that I did and contributed to was by helping with creating the samples, cell splitting and counting, and also running lab tests to see if our compound was successful. So the end results I had at this program were, when running a Western blot and quantifying them to be normalized to beta-actin, all of our samples came back with no significant difference. 3 questions I had at the end of the program were: 1, how could our results have changed if we used different samples? 2, if RIPK1 and MLKL were to bind, what outcome would occur? And 3, would there be a difference in results if a different visualization from Western blots was used? So here I am doing some cell splitting, and I am just adding trypsin and media to cells, and then I'm going to separate them into 2 flasks, and then from then I will start the process of cell counting. So this is where I start the process of cell counting. I am just grabbing my cells, some water, and the blue liquid, which I will pipette into a hemoglass tray and place it under a microscope to count all my cells and then do a quick equation to find the total. So in the next 2 clips, I am just adding my more media, and the cells into some wells, and then from then on, that's when I do some of my cell counting. And in this final clip, I'm showing the cells and what they look like under the microscope while I'm just trying to count to find out the total, including the equation.